Okay, right, let's start chapter 6, Control and Utilization of Metal Ion Concentration in Cells. In the chapter 5, we studied how the biology can select certain metals and how they can uptake and store. Here we will more focus in the deeper side, the metal ion concentration, how the concentration can be regulated inside the biology and what the importance of concentration of certain metal is. Okay, the first section is beneficial and toxic effects of metal ions. Metal ion concentration must be maintained within the proper ranges in cells. If the concentration is too low, the organism can suffer from metal ion deficiency. You know that some metal ions act an important role in catalyst, electron transfer, oxygen transfer, etc. If there is deficient of metals, those kinds of specific metabolic actions cannot occur. Above or lower threshold, then ion can fulfill its biological functions. But above the upper threshold, the effects of metal ion toxicity will arise. For example, if a metal ion binds at an inappropriate site, it might compete with other beneficial metal ions. Or there might be undesirable reactivity of the metal ion when it is not properly controlled in its normal binding site. For example, iron Fe is important in biology, but if there are too much ions in the biology, the ion will be bound to different kinds of ligands which are not intended. It can compete with zinc ions in zinc finger, and it can compete with zinc ion in carbonic anhydride. So they can interfere the action of other metal ions. So the metal concentration should reside in a proper concentration here. If the metal concentration is um, too low for in terms of beneficial metal ions, the cell growth or organismal health is very low. But within this concentration range, their action is good, and the cell growth and metabolic actions comes in desirable way. But above a certain metal concentration, it comes down. This graph is applied to every kinds of metals. Only the toxic metal ions, this curve shifts. So when the toxic metal ions concentration is low, it is not that toxic, but when it increases a little bit, it competes with other metals. So the ions are an example of beneficial metal, and mercury and cadmium, uh, those kinds of heavy metals are the examples of toxic metal ions. Remember the wording homeostasis. Homeostasis means the um, control of certain range. So it includes the maintenance of the concentration of beneficial metal ions in the correct range and detoxification, the removal of toxic concentration of non-beneficial metal ions. So the homeostasis requires a balance between metal ion uptake, utilization, storage, and excretion. Okay, two examples of concentration issue. The first one is iron. It acts as oxygen transport system and many different kinds of metabolism. It participates in a variety of electron transport pathways. So iron is a beneficial metal. In case of deficiency, it produces anemia. Yeah, it's not desirable condition. And in overload condition is also not desirable. Excess ion can accumulate and is not easily excreted. And excess amounts of the metal ions which are released into the system can catalyze the formation of various oxygen-based free radicals and extensively damage tissue. Excessive amount of ion makes oxygen-based free radicals and damage tissue. Even though iron is a beneficial metal, it can give toxic effect when the concentration range is not in an appropriate region. To avoid these effects, drugs such as desferrioxamine are given to chelate the iron and facilitate its excretion. So, in case of overload iron, we have to excrete the overload ion by utilizing chelate. Another example is the toxic effects of metal ion. Cisplatin. You know that cisplatin is a kind of drug, anti-cancer drug. The cisplatin DNA adducts are harmful. It may because the cisplatin can block the transcription of DNA. So it's harmful, but it means that it is harmful for both normal and tumor tissues. But the toxic effect is more severe to tumor than normal tissue. The reason is simple. Tumor tissue doubles faster than the normal tissues. It means that the doubling time of cancer cell is much shorter than the normal cells. In real, the cancer cells doubles in 24 hours, but the normal cells like fibroblast cells requires more than 72 hours to double. So, so the DNA transcription is more important in tumor cells. So the formation of cisplatin DNA adduct is more harmful to tumor. So uh, it results in a kind of beneficial effect at the organismal level. For your information, there exists a similar compounds, diamine dichloroplatinum. Cisplatin is platinum here. These compounds can also be utilized as the formation of DNA adducts, but it undergoes ligand substitution reaction at a rate 10 power 5 times more rapidly than cisplatin. So it is much toxic. That's why palladium complex is not utilized as an anti-cancer. Palladium complex reacts rapidly with many biological targets that it can damage or inactivate before reversibly damaging DNA in the nucleus of cells. The regulation. We will see the example of iron. Focus on the system involved in the regulation of ion immobilization and storage. We will study ion transport protein and the storage protein. We have already seen the example of storage protein, the name of ferritin. And here we will again study the transport protein named transferrin. Let's start solubilization, uptake, and transport of iron, Fe3+. Iron 3+ is quite insoluble at neutral pH. That's the problem. It's not insoluble. At neutral pH, I mean in the biological, physiological pH. Even though iron may be abundant in the environment, very low levels of the hydrated iron will be present in solution. So only a small quantity can be utilized. So bacteria produces siderophore like enterbactin to chelate iron and solubilize it. Different from bacteria, mammals do not utilize chelate. They utilize proteins. The proteins of the transferrin family, glycoproteins, tightly bind to Fe3+ ion. These proteins allow solubilization and transport of ion under conditions where the free ion would be hopelessly insoluble. So the transferrin is a kind of transport system like in Sidro4. The difference between transferrin and Sidro4 is the size and functions. Transferrin is a protein, so the size is much bigger than Sidro4. But transferrin can be recycled, while the Sidro4, like enterobactin, is produced and is dissolved, broken after the transport of ion. So it seems that transferrin lies in the higher level of metabolism. Okay, the X-ray crystal structure of proteins consists of the protein transferrin. It consists of two similar domains, each of which binds one ion atoms. Each domain consists of two subdomains that bend like a hinge when bound to ion. That is, the subdomain clamp down on the ion carbonate. This is the structure of lactoferrin as a kind of transferrin. Here you can see two red spots. This is ion, and this is one unit, and this is another unit. Each unit can contain one ion, so it means that one transferrin can accommodate two ions. And let's look at the coordination structure around the ion. So this is the structure of one subunit here, one subunit of apotransferrin. Transferrin without ion. This is the structure of transferrin with ion. Can you catch the difference? Here, this part is, the space is quite large, but here it comes smaller. So it's a subdomain clamped down structure. Here, there exists some amino acid residues to coordinate to ion, so these two parts hinge down to clamp the ion. The structure, the coordination structure in detail. It is stabilized in the ion is stabilized in the distorted octahedral state. Octahedral state, so there exist 6 ligands. There are 2 phenolate oxygen. 2 phenolates are from the tyrosine amino acid. 1 imidazole nitrogen from histidine amino acid. 1 carboxylate oxygen from aspartic acid. So 1, 2, 3, 4 ligands are donated by the protein, and the other 2 oxygens are from the bound carbonate. The carbonate is also hydrogen bonded to the side chain from the arginine and threonine of the amino acid. So the ion is really tightly bound to the protein. Look at the structure. Here there is ion, and as I told you, there are 4 ligands from the amino acids: tyrosine, histidine, the phenolate, imidazole, phenolate, and one more imidazole here. No. One more carboxylate here. And the 2 coordinations are from the carbonate anion. And this carbonate anion is also bound to the amino acid residues through hydrogen bond. So ion is really tightly bound. The protein apparently has a binding pocket for carbonate that consists of hydrogen bonding units contributed by side chains. An ion 3 plus can now coordinate in an octahedral site. Once the transferrin catch the ion, then they have to find the route to enter the cells. That route is called receptor-mediated endocytosis. Remember the case of the Sidro4 enterobactin. Enterobactin coordinated to ion. Then they get through the cellular membrane to enter the cells. That one is a kind of diffusion, not very efficient one. But this one The receptor-mediated endocytosis is a concrete way to enter the cells. Transferrin, the protein, binds to a membrane receptor, and then the cell recognizes that the transferrins are at the surface, and they engulf the transferrin by endocytosis to form so-called coated vesicles. Here, this is the schematic diagram for the endocytosis and the recycling of transferrin. The vesicle coat is a protein called clathrin. Here, once this is transferrin with ion, and they are recognized by the receptor here. Once they are attached to the receptor, the membrane pinch it off to make the vesicle. To make vesicle, the protein, which is called clathrin, is coating the inside of the membrane. Then it makes vesicle. And the vesicle is called endosome. Inside the endosome, the pH decrease due to the proton pump. The proton pump and the acidic media inside the endosome facilitate the release of ion, and then the ion is freely released from the transferrin. Then the ion is released into the cells from the endosome to the cells. Then the apotransferrin is excreted out of the cells for the recycling. So the released ion is then available for use for storage in ferritin. And a portion of the vesicle containing the receptors that have apotransferrin still bound then fuses with the plasma membrane. Upon being re-exposed to the outside of the cell with pH near 7.4, the apotransferrin is released, and then it repeats itself. So the process is somewhat similar to the siderophore, but the transferrin is more effective in terms of cellular entrance and the recycling. Now we will see how the uptake and storage is regulated. Organisms have also developed methods for accumulating and storing metal ions for later use. The molecular details of how the entry and departure of ion from ferritin are modulated by cells are not yet completely understood, but the mechanism by which ion controls the genes that produce the proteins required for siderophore production as well as for the synthesis of transferrin or ferritin. So when the transferrin receptor is expressed, it means then that the cells are ready to take up the ion, and when the ferritin apoprotein is produced, it means that the cells are ready to store the ion. So when the ion concentration level goes down, the cells produces transferrin receptor, and when the ion level concentration goes too high, the cells develop ferritin apoprotein for the storage of ions. The genes involved in siderophore biosynthesis in bacteria are regulated by ion at the transcription. So let's look at that transcription level synthesis. There is an two system when the ion concentration is high and low. When the ion concentration is high, FUR protein, FUR is an ion uptake regulator. This protein contains bound metal ion and binds to specific DNA regulatory sequences. So this can repress the transcription of the relevant genes. So when the ion level is very high, the transcription of C04 is regulated. And when the metal ion concentration is low, the FUR protein does not bind metal ions, does not bind to DNA, so it does not regulate the transcription, so the transcription can occur. The stored ferritin mRNA is translated relatively slowly if cellular ion levels are low. Cellular ion level low, it means that there is less necessity to store ions, so that the ferritin mRNA translation is low. As the cellular ion levels increase, and there is necessity to store ions, so the rate of ferritin transcription increases dramatically. So the many ferritin proteins, alpha proteins are produced and store the excess amount of iron. In case of transferrin, the transferrin receptor is relatively unstable under ion-rich conditions. So when ion is very high in concentration, the transferrin receptor transcription is regulated. But when the ion concentration is low, the mRNA for the transferrin receptor became stable, so the transferrin receptor is expressed. These two phenomena are mediated by similar sequence structures in the mRNA molecule, the structure that have been termed ion-responsive elements, IREs. The IREs act as binding sites for a common protein called the IRE binding protein. When the IRE binding protein binds to the ferritin mRNA, it prevents translation of the message into protein. So the expression does not occur well. And when it binds to the transferrin receptor mRNA, it inhibits degradation of the RNA, so the translation is allowed. Look at the structure. There is a mRNA region. At high levels of ion, the clusters in the protein, the relatively low affinity for IRE containing mRNA molecules, and under low ion concentration, the cluster is dissociated, so there is a change of increasing the affinity to RNA. So, or is there is a certain unit which can sense the concentration of ion, and that sensor regulates the transcription of mRNA. And finally, the translation, the expression of transferrin or ferritin is regulated.